Thank you very much, and good afternoon, everyone. As an HFSP fellow, I have had the enormous privilege to be studying one of the most remarkable biological processes that gave rise to the life of each and every one of us. And what I'm talking about is the process of fertilization depicted here in this image. And just to convince you how remarkable this process is, this is a movie of fertilization in starfish, where you can see that a very small and motile cell, the sperm, can interact with this large and immotile cell, the egg, to give rise to the life of most sexually reproducing organisms. And so I study this process, and the main question I'm trying to address is how is this process achieved at a molecular level? So there's a handful of factors that we already know are important for this process, which are depicted here. But we still have no idea how these factors are working together to mediate gamete binding and fusion. So we study the zebrafish as a model system to study vertebrate fertilization, and that is because it happens externally. We can collect a large number of gametes for biochemical assays, and this process can be imaged as well. So to cut a long story short, we have, through an alphafold-multimer screen, discovered a protein complex that sits on the surface of the sperm and is important for fertilization. It's composed of two previously known proteins, Izuma-1 and SPACA6, and a newly identified factor, TMEMD1, which we also showed that is essential for fertilization in both fish and mammals. And to make the story even more interesting, what we found was that this conserved sperm complex interacts with different receptors on the egg in zebrafish and in mammals, suggesting that somehow these proteins evolved to be able to interact with different factors on the egg. And so if you're interested in protein-protein interactions or in how evolution gave rise to this very fascinating way to connect sperm and egg, please visit me at my poster number 37. Thank you.
Thank you.